Okay. Okay. Hi, everyone. My name is Etchi, and I am a third-year bioengineering student here at UCR, and my project is entitled The Downregulation of Genes by Neuregulin-1 in PMCAO Stroke Models. And for this project, I'm working under Dr. Victor Rogers from the Department of Bioengineering and Dr. Byron Ford from the Division of Biomedical Sciences. And for this project, my main goal is to investigate the growth factor Neuregulin-1 as a potential treatment for stroke. Stroke is the 5th leading cause of death in the United States and the leading cause of long-term disability. And ischemic stroke occurs when blood flow to the brain becomes blocked. And when blood flow to the brain becomes blocked, this can inhibit the transport of important nutrients such as oxygen to the brain. And in this image to the upper left, we can see that we have a graphical representation of an ischemic stroke. So we have our blood vessel, and then we have an obstruction. And we can see that this obstruction is preventing the transport of important nutrients to the brain. And when our brain can't get these important nutrients, we can end up having situations such as apoptosis, which is programmed cell death. And we can also have a number of neurological deficits as mentioned earlier. And currently, treatment for stroke is limited. And the main treatment option for stroke is something called TPA, which stands for tissue plasminogen activator. But the catch with this treatment is that only about 3 to 5% of all stroke patients actually qualify for this treatment. And this is due to a variety of reasons. For example, in order to actually qualify for TPA, you need to come in to the hospital within 4.5 hours of the initial stroke. And majority of patients come in way after this. And depending on the patient's medical history, such as other conditions they may have or other medications they're taking, TPA might not be the suitable option. So because of this, our lab recognizes that there is a dire need for new treatment methods. And this is where Neuregulin-1 comes in. And Neuregulin-1 is a growth factor that has been studied for its neuroprotective properties. Neuregulin-1 has been shown to protect against ischemia-induced brain injury and prevent apoptosis from stroke when administered before the onset of ischemia. And another great thing about Neuregulin-1 is that it has a 12-hour time window. So compared to TPA, Neuregulin-1 can really be an amazing treatment if everything works out well. And for this project, my main goal was to analyze the gene expression of rats that were first treated with Neuregulin-1, and then they underwent permanent middle cerebral artery occlusion, also known as PMCAO. And this is just another way of saying stroke. And this method is just when we insert a filament into the middle cerebral artery of our rats. And by doing this, we are obstructing blood flow. And as a result, it's mimicking an ischemic stroke. And to do our analysis, we primarily use the transcriptome analysis console, also known as TAC. And this way we use mRNA microarrays to examine the gene regulation at the sham control group and the PMCAO, which is basically our stroke level, and then also the Neuregulin-1 treatment conditions. And the way these microarrays work, there's a chip and there's many probes on it. And when we have our sample from the rat brain, it will bind to the corresponding probes on the chip. And additionally, we also use something called string to analyze the connections between the genes. And now moving on to the analysis. So when we started with this project, we had about 30,000 total genes that were provided by TAC. And significant genes were selected by a p-value of less than 5% and a fold change of 1.5 or higher. And in order to narrow down my selection of genes I wanted to focus on, I decided to focus on genes that had a pattern of upregulation with MCAO and downregulation with Neuregulin-1. And for this presentation, I decided to feature 3 genes of interest: BDNF, which is a brain-derived neurotropic factor, TGF-B1, which is a transforming growth factor, and NR4A1, which is a nuclear receptor. And just to elaborate more on what I mean by these patterns, so for example, as you can see here in this middle column, all these genes of interest seem to follow a similar pattern. So first we have our sham, which is our control group, and this is at one expression level. And then when we look at MCAO, we notice that there is an increase of expression. So when I say upregulated, I mean upregulated with respect to sham. And then I noticed that with the neuregulin 1 group, there is a decrease of expression level when compared to MCAO, which is our stroke. So my main focus is really to look at, is there any sort of similarities or commonalities with genes that seem to follow this pattern, or more specifically, genes that were downregulated with neuregulin 1 treatment. And after doing some literature analysis and trying to understand more about this pattern, it seems that overexpression of these genes can actually lead to apoptosis. And then next, what I did to continue further analysis, I entered my genes of interest into something called String, which is a database that can help me see known and predicted protein interactions within my genes. And then what I did, I decided to add the next 5 closest interactions to see if I can find any shared pathways within all these genes. And as we can see here, I have my 3 genes of interest, which are highlighted in this green box. And then next I have my other 5 closest interactions. And for the colors of these genes, which are represented by these orbs, these colors were just given randomly, so the colors don't really indicate anything. But then another thing we notice is that we have different types of connections, and the colors of these connections actually have significance. So for example, this light lime green connections between my genes of interest, this represents text mining. And this just means that these genes may have all come up together in a paper or other literature. And then next, in our clusters, for example, I'll just focus on this bottom one right here. We can see that there's various colors. So it might be a little bit hard to see, but there's a purple, and that purple just means it's coming from a known database. And then there's also a little bit of blue, and that blue just means that it's coming from curated bases. So both blue and purple connections in my analysis suggest that there is high confidence in these connections. But then when we look at the text mining, for example, and also there is a black connection in here, which represents co-expression. These are just predicted connections, and the confidence in these connections are not as high. And then another thing I was able to do with String is to see if there were any shared pathways between all of these genes. So first I noticed that there was the MAPK pathway, which stands for mitogen activated protein kinases. And in this pathway, actually some of these genes, depending on the stimuli, they can actually influence apoptotic responses. And then next I noticed FOXO signaling. And in this pathway, it's mainly a group of transcription factors that can also play a role in apoptosis. And then I also noticed the TGF-beta signaling, Hippo signaling, and PI3K-AKT signaling. And just basically looking at my initial findings, I was able to come up with my preliminary results, which suggest that these pathways may actually amplify apoptotic responses. And then, so moving on to my conclusions and future work. So my preliminary results suggest that neuregulin 1 can actually downregulate genes associated with pathways that not only increase vascular permeability, but also promote apoptosis. And then future studies will include investigating neuregulin 1 as a treatment for stroke in humans. And since my preliminary results are suggesting that these genes that follow this expression pattern might actually promote apoptosis, in the future, maybe we can conduct more studies to target these genes, or more specifically, these pathways, so we can essentially kill 2 birds with 1 stone. And I would just like to thank my PIs, Dr. Rogers and Dr. Ford, for all their support, as well as the B2K group and the Ford Lab. And I would also like to give a special thanks to the NIH and the Mark U Star program for funding this project. Thank you for your time. Any questions?
Excellent job. Thank you, Etchi.